for today, and in this video we're going to discuss the history of cell. We'll talk about who the scientists led to the development of cell, we'll talk about their history, we'll talk about the cell theory, and we'll talk about the kind of cell, such as eukaryotes and prokaryotes, and we'll talk about the diversity of cell. So let's go and get started. So the first thing I want to mention is that when the history started or began, it all began in 1590, where the two Dutch spectacles, Hans and Zacharias Janssen, took the credit of making compound microscope. However, this microscope could not gain popularity as they failed to increase the resolution and therefore magnify images appear blurry as a result of no significant scientific invention came from them. Over about 100 years, over a period of time, significant improvements were made to this microscope. And one thing to mention is that all life is made from cell, and keep in mind that cell itself wasn't even discovered until 1600s with the invention of the microscope. And here we have a picture of the microscope that actually discovered cells. This was a microscope that belonged to Robert Hooke. In 1665, Robert Hooke was one of those scientists who improved the basic design of the existing compound microscope. He constructed a microscope with three lenses to enlarge the object and a stage light to illuminate the object. Robert Hooke first observed cork, which happened to be dead bark tree. Cork is the bark of type of oak tree. Here we have an example there, a picture of cork. And when Robert Hooke placed a slice of cork under his microscope, he observed some wonderful structure that he had never seen before. To him, the cork appeared as if it was made of tiny rooms that monks live in monastery. This room in Latin known as cellula, therefore he called them cells. And in 1673, after Robert Hooke observations, a Dutch textile merchant, Anton van Leeuwenhoek, had observed living cells under his self-made microscope. And here we have a picture of the microscope that Leeuwenhoek used to observe organisms. And Leeuwenhoek had a simple magnifier that checked the quality of fabric, and his microscope could magnify an object by around 300 times. And Leeuwenhoek did not know what these tiny cells were. What he actually observed under his microscope was bacteria and protozoa, and he called them animalcules. Who developed the cell theory? In 1838, a German botanist named Matthias Leiden worked on microscope observation of plant tissues. And here we have a picture of Matthias Leiden, and Matthias Leiden concluded that all plants were made of cells. And in 1839, a German geologist named Theodor Swan has made similar observation in animal tissues. And here we have a picture of Theodor Swan, and Theodor Swan concluded that all animals were made of cells. And Swan also concluded that all life is composed of cells. Swan also a co-founder of the cell theory. In 1839, both have compared their observation and found similarities between plants and animal tissues, and they finally concluded that cells are the basic structural unit of plants and animals. Even though Swan and Leiden came together to formulate their cell theory, they disagreed on one thing. Swan did not agree with Leiden's statement that cells form spontaneously. However, Leiden's statement wasn't in fact wrong, because soon after the medical doctor Rudolf Virchow in 1855 proved that cells came from pre-existing cells, which simply means that new cells were formed from old cells. What is cell theory? Cell theory is a universally accepted scientific theory which mainly states three basic parts. And cell theory we state that all living things are composed of one or more cells, and cells are organism basic units of structure and function, and cells came only from existing cells. And here we have a picture of cells, and you can see a few part levels like lysosome, nucleus, mitochondria, Golgi body, and smooth and rough ER. Number of cells. Although all living things are made of cells, organism may be unicellular, composed of one cells, or multicellular, composed of many cells that may organize into tissues. So next, I'd like to go into the diversity of cells, and here we have a three-part of cell diversity, such as size, shape, and internal organization. Here we have a shape of diversity of cells. Cells differ widely in shape. Most cells are roughly cuboidal or spherical. And one thing I want you to know is that the shape of cell really depends upon its function, because not all animals' cells are round and not every plant cells are rectangular shape. And here we have a picture of cell diversity size. We have the smallest cells. The bacterium have 2 to 10 millimeters, and the longest cells, giraffe nerve cells, total length up to 2 meters, and the biggest cells, ostrich egg, have a 6 inches long and 5 inches wide. And cell diversity internal organization, nucleus contain DNA which direct the activity of the cells. Organelles, a cell's component that performs a specific function in the cells. Eukaryotes, cells that contain a nucleus and membrane-bound organelles. And prokaryotes, cells that lack of nuclei and membrane-bound organelles. And here we have a picture of cells known as eukaryotes. And eukaryote is a simple cell organism that lack of nucleus and membrane-bound organelles. And the main thing to keep in mind is that prokaryotic cells are not divided upon inside by membrane wall. And nucleate found in the center region of the cells and surrounded by cell membrane and cell wall, made out of peptidoglycan. A polymer composed of linked carbohydrates and small proteins, and contain ribosomes, no membrane, in their cytoplasm to make proteins. And here we have another picture of prokaryotic cells. And we have ribosomes. Ribosomes are molecular machines that synthesize proteins. And you have capsule. Capsule is a sticky and help the cells attach to its surface and its environment. And capsule also have an outermost layer of carbohydrates. And we have the cell wall. Cell wall provide an extra layer of protection, help the cells maintain its shape and prevent dehydration. And you have the plasma membrane. Plasma membrane is an outer covering the cell's interior from its surrounding environment. And you have the DNA. DNA is the genetic materials of the cells. And lastly, we have a flagella. Flagella are the structure that act as a rotary motor to help the bacteria move. And here we have the structure and function of organelles. The structure and function of the following organelles will be discussed. We'll have a cell membrane, nucleus, a cell wall, cytoplasm, cytoskeleton, ribosomes, endoplasmic reticulum, Golgi apparatus, mitochondria, lysosome, peroxisome, cilia and flagella, basal body, centrioles, vacuoles, and plastids. Cell membrane structure phospholipid bilayer with proteins that function as channels, markers, and receptors. Also contain cholesterols which provide rigidity, function selectively permeable, boundary between the cells and external environment. Cell membrane, also called plasma membrane, and plasma membrane or cell membrane provide protection for cells It also provides a fixed environment inside of the cells, and that membrane has several functions. One is that transport toxic substances outside of the cells, and another is that plasma membrane have proteins on it which interact with one another cells, and those proteins can be glycoproteins, means there's a sugar, fats, and proteins, and those proteins which stick outside of the plasma membrane will allow one cells to interact with one another. And the cell membrane also provide some structural support for a cells.